Hello, my dear students. So, now we are going to start a new chapter from Science second of Standard 10. Okay, so from part 2, we are going to start with the very first chapter. Okay, in this particular part of Science and Technology, that is in part 2, we mainly deal with the knowledge which belongs to the botany and zoology section. Okay, so in short, we study about life, okay, and living organisms in this particular part of the subject. So to begin with, as we are going to start with the very first chapter, the name of the chapter is Heredity and Evolution. So in this particular lesson, we are going to see how the characters of parent is transferred to the offspring. Okay, how the living organisms have of same species have same characteristics, and we will see the evolution of life on the planet Earth. Okay, so there are several theories which are given in respect to this particular topic, that is evolution. We will deal those topic in this following lecture. Now, before going to the chapter, let us answer this basic questions. So what is the first question over here? It is, which component of the cellular nucleus of living organisms carries hereditary characters? Okay, so what is the question? Which is the component in our cell that is going to carry the characteristics of parents to the offspring, to their children? Okay, so what is that component? It is DNA. What is that component? It is DNA. And we all know the formula, the full form of this particular abbreviation, that is DNA. The DNA stands for deoxyribonucleic acid. DNA stands for deoxyribonucleic acid. Okay, so this DNA is responsible for the transfer of characteristic from parents to the offspring. Then second question, what do we call to the process of transfer of physical and mental characters from parents to the progeny? Progeny means offspring, that is children. Okay, so what is the name of this process where the characters are transferred from parents to the offspring? This process is called as heredity. It is called as heredity process. Okay, then third question, which is again a very important question, mark this as important. Okay. So what is the question number three? Which are the components that in the DNA molecule? Okay. So what components are there in the DNA molecule? Okay. So DNA is made up of double-stranded sugar bonds. Okay, double-stranded sugar like thread. Okay, it is made up of this. This is the structure of DNA. Okay, and this there are two strands. Okay, made up of sugar, and in between these two strands there are four more components of the DNA. What are the components of DNA? First, thymine, then adenine, then cytosine, and then guanine. Okay, so there are four components between the strands of a DNA molecule. Those are thymine, adenine, guanine, and cytosine. Okay, there is one more molecule which is there in cell, that is RNA. Okay, we know about DNA. There is also RNA, which is ribonucleic acid. Okay, so in RNA, thymine is not present. Instead of thymine, you will find uracil. Okay. Now we will see the heredity and hereditary changes. So what is heredity? We are going to discuss this in this particular lecture. You know that heredity is the transfer of biological characters from one generation to another via genes. Okay, so this is the definition of heredity. Heredity is the transfer of biological characters from one generation to another via genes. Okay, so remember this. So पेरेंट्स से ऑफस्प्रिंग में जो कैरेक्टर्स ट्रांसफर होते हैं उस प्रोसेस को हम लोग क्या बोलते हैं, हेरिडिटी बोलते हैं. Now, the man who had discovered this particular concept, heredity, and who had contributed in a very great way in the knowledge of genetics, the name of that man is Gregor Johann Mandel. Okay, so he is known as the pioneer of the modern genetics. Okay, that is Gregor Johann Mandel is called as the father of modern genetics. Okay. Now, it took a long time to understand the conclusions of his research about heredity. So, whatever Gregor Johann Mandel has researched, okay, so his research were concluded after a very long time. उसने जो रिसर्च किया था वो रिसर्च को समझने को बहुत लंबा समय गया, क्योंकि जोहन मेंडल जो था वो अपने टाइम में बहुत आगे था. ग्रेगोर जोहन मेंडल जो था वो अपने टाइम के हिसाब से बहुत ही आगे था इन टर्म्स ऑफ रिसर्च इन जेनेटिक्स. ओके, तो बहुत टाइम के बाद जो बाकी साइंटिस्ट थे उन्हें समझ में आया कि ग्रेगोर जोहन मेंडल ने क्या लिखा है. ओके, उसका क्या रिसर्च है, क्या कंक्लूजन है उस रिसर्च का. In 1901, the reasons behind the sudden changes were understood due to the mutational theory of eukaryotes. Okay, जो म्यूटेशनल चेंजेस होते हैं पेरेंट्स और ऑफस्प्रिंग 100% सेम नहीं होते, कुछ ना कुछ डिफरेंस तो होता है. और वो डिफरेंस के पीछे क्या रीजन था, वो 1901 इन द ईयर 1901 एक साइंटिस्ट है, एक जूलॉजिस्ट है जिसका नाम है यूगो देवरेस, उसने क्या किया, फाइंड किया. Meanwhile, in 1902, Walter Sutton observed the paired chromosomes in the cells of grasshopper. Okay, this is again a very important statement. वॉल्टर सटन नाम के एक साइंटिस्ट ने 1902 में सबसे पहले क्रोमोसोम्स को डिस्कवर किया. ओके, और वो क्रोमोसोम्स किसमें डिस्कवर हुए थे, ग्रास हॉपर में. और क्रोमोसोम्स क्या है, इट आर दोज आर द कंपोनेंट्स प्रेजेंट इन द डीएनए. ओके, sorry, those are the components present in the cells of a living organism. Until then it was not known to anyone. उसके पहले 1902 के पहले किसी को भी क्रोमोसोम्स के बारे में मालूम नहीं था. Research started in the direction of finding the nature of genetic material when it was proved that genes are carried via chromosomes. देखो, जो हमारा डीएनए मॉलिक्यूल होता है, जो जीन जो होता है, वो कौन कैरी करता है, वो कौन ट्रांसफर करता है, क्रोमोसोम ट्रांसफर करता है. कौन कैरी करता है, वो क्रोमोसोम्स ट्रांसफर करता है. ओके, तो ह्यूमन बींग्स में टोटल कितने, एक सेल में कितने क्रोमोसोम्स होते हैं, 46 क्रोमोसोम्स होते हैं. ओके, ह्यूमन बींग्स में एक सेल में कितने क्रोमोसोम्स होते हैं, 46 क्रोमोसोम्स होते हैं. उसमें से 22 पेयर्स 22 pairs of chromosomes are autosomes. Okay, 22 pairs of chromosomes are called as autosomes, and one pair of chromosome is called as sex chromosome. It is called as sex chromosome. Okay, so we are going to deal with this part also, chromosomes, in the foremost lectures. Okay, now through which 1944 trio of scientists. Now there are three scientists: Oswald Avery, McLean McCarthy, and Colin MacLeod. Okay, these three scientists have proved that except viruses, all the living organisms have DNA as genetic material. जितने भी लिविंग ऑर्गेनिजम्स है एक्सेप्ट वायरस, उन सभी के पास जेनेटिक मटेरियल के तौर पे है कौन सा कंपोनेंट है, डीएनए है. सो ईच एंड एवरी लिविंग ऑर्गेनिज्म हैव डीएनए एज अ जेनेटिक material. Okay, but viruses do not have DNA as genetic material. In viruses you will find RNA. Okay, RNA as genetic material in viruses. Okay, now in 1961 the French genetist Francois Jacob and Jack Monod. Okay, there are two geneticists. Okay, Francois Jacob and Jack Monod. They proposed a model for process of protein synthesis with the help of DNA in bacterial cells. उन्होंने क्या प्रूफ किया, क्या फाइंड किया कि डीएनए जो है वो प्रोटीन को सिंथेसाइज करने के लिए, प्रोटीन को बनाने के लिए हेल्प करता है। ओके, हमारे सेल्स को it helped to uncover genetic codes hidden in DNA. And due to this discovery, the scientists have discovered the genetic codes. Okay, genetic codes is nothing but the sequence of these four: thymine, adenine, cytosine, and guanine. Okay, in the DNA molecule. Thereby, the technique of recombinant DNA technology emerged, which has vast scope in the field of genetic engineering. So, such discoveries have led to new discoveries, and this new discoveries has improved the lifestyle of human beings. It has affected, okay, or it has affected in a good way in the field of technology. Okay, so new discoveries are being made every day. The science of heredity is useful for diagnosis, treatment, and prevention of hereditary disorders, production of hybrid varieties of animals and plants, okay, and in industrial processes in which microbes are used. सो हम लोग हेरिडिटी या फिर हेरिडेटरी साइंस को स्टडी क्यों कर रहे हैं? व्हाट इज द रीजन? बिकॉज इट इज यूजफुल फॉर अस इन द डायग्नोसिस, ट्रीटमेंट एंड प्रिवेंशन ऑफ हेरिडेटरी डिसऑर्डर्स। ऐसे डिसऑर्डर्स जो पेरेंट्स की वजह से ऑफस्प्रिंग को होते हैं, उन डिसऑर्डर्स को क्योर करने के लिए हम लोग को स्टडी करना जरूरी है हेरिडिटी को। उसके बाद नए हाइब्रिड वैरायटीज एनिमल्स के और प्लांट्स के बनाने के लिए, प्रोड्यूस करने के लिए भी हमें हेरिडिटी की स्टडी करनी है। और बहुत सारे ऐसे इंडस्ट्रियल प्रोसेसेस हैं जहाँ पे हम लोग माइक्रो ऑर्गेनिजम्स का इस्तेमाल करते हैं प्रोडक्ट को प्रोड्यूस करने के लिए, तो वहाँ पे भी हमें हेरिडिटी की नॉलेज होनी चाहिए। ओके, so this is about the heredity. In this particular lecture, we have seen the introduction to this chapter. Okay. In the next lecture, we will see about transcription, translation, and translocation. Okay. Until then.